Hello, it's my pleasure to welcome you to this virtual scientific session developed by the American Neurogastroenterology and Motility Society, focusing on basic concepts in neurogastroenterology. My name is Beverly Greenwood van Meerveld, and I'm the immediate past president of the ANMS and the current director of the ANMS Institute. The topic of today's ANMS Symposia is GI dysmotility, the role of non-neuronal cells, and we will have 3 presentations from experts on this exciting topic. But before we start, I'd like to personally thank each of our 3 speakers today for taking the time out of their busy schedules to join us. I would also like to thank all the members of the ANMS Meetings Task Force for developing this virtual program that will continue at least through mid-2021. And our next virtual scientific session will be in February, so please save the date. It's now my pleasure to introduce today's moderator, Dr. Keith Sharkey. Keith is a professor of physiology and pharmacology at the University of Calgary. He's also the Crohn's Colitis Canada Chair in IBD Research. Dr. Sharkey studies the neural control of the GI tract and brain-gut interactions in health and disease. Thank you, Dr. Sharkey.
Well, thank you, Beverly, for that kind introduction. It's really a pleasure to be here today and to moderate this session. It's a really exciting session, and we're very lucky to have 3 amazing speakers. So without further ado, I'm going to introduce the speakers, and then at the end of the 3 talks, I'm going to moderate a question and answer session. Please put your questions in the question and answer box, not the chat box at the bottom of the Zoom link. So without further ado, I want to welcome Meena Rao. Meena's a pediatric gastroenterologist directing a research program focused on functional interactions between neurons and glial cells in the enteric nervous system and how disruption of these contribute to digestive diseases. She received her undergraduate training at WashU. She has a neuroscience graduate degree, a PhD from Johns Hopkins, and she's completed residency training in peds and a fellowship in pediatric gastroenterology, hepatology, and nutrition at Boston Children's. She went back to Boston in 2018 and has a lab there at Boston Children's and Harvard. Meena's going to talk to us about emerging roles of enteric glia in neurogastroenterology and motility disorders. So without further ado, I welcome Dr. Rao.
Thank you, Keith, for that very kind introduction, as well as the organizers for the opportunity to present today. Here are my disclosures, none of which are directly relevant to the talk today. So as most of the audience knows, the digestive tract is endowed with a large complex component of the peripheral nervous system called the enteric nervous system, or ENS. And this ENS is pretty critical for almost every aspect of gut function, most famously regulating gut motility. So one can imagine when there's such a critically important system in place that when it's not working, it can play a role in causing disease. And the most famous of these diseases are disorders in which segments of the gut are actually missing part of the enteric nervous system, such as Hirschsprung's disease or Chagas disease. But there also is an increasing appreciation that there are a wide range of both functional and inflammatory GI disorders, as well as neurologic disorders in which the enteric nervous system is present, but just not functioning as it should. The neurons of the enteric nervous system are organized into collections or ganglia that are located in 2 main regions, the myenteric ganglia in the muscularis externa of the gut and the submucosal ganglia in the submucosa. Now, the neurons are actually vastly outnumbered by a large population of non-neuronal cells called glial cells that are found within these ganglia, but are also distributed much more widely through the lamina of the gut. So to get a sense of this, we'll start with the muscle layers. Here in the muscularis externa, you can see that there's large populations of green glial cells organized into ganglia surrounding neurons. These are the myenteric glia, and these are by far the most studied of all the glial cells in the gut. There also is this beautiful array of intramuscular glia that's found throughout the muscularis externa along nerve fibers, and these are glia with central cell bodies and bipolar processes. These are intramuscular glia. As we moved a little bit closer towards the lumen of the gut, one would reach the mucosa. And here we would find either the bases of crypts in the colon or intestinal epithelial crypts in the small intestine. And all of these crypts are surrounded by glia and their processes. And as we move even closer towards the lumen in the small intestine, we would reach the villi. As one can see here, every single villus in the small intestine of both mouse and humans has several glial cells that are arborizing these extensive processes that are interacting with blood vessels, immune cells, microbiome, microbial products, as well as specialized epithelial cells such as enteroendocrine cells. So despite the fact that these enteric glia actually represent the largest population of glial cells outside of the brain, and they really form this just remarkable three-dimensional network that extends throughout the layers of the gut, we're really at the tip of the iceberg in really understanding what they normally do and what their roles could be in digestive disease. Now, some of the work that really laid the groundwork for understanding what their potential functions might be started about 20 years ago with this work by Bush and colleagues that was published in Cell. And they reasoned that one way to understand what these cells might be doing is to actually get rid of them from the GI tract and then see what happens to gut functions. And they did this by engineering mice in which a glial promoter drove expression of a viral enzyme called thymidine kinase. And when thymidine kinase sees its substrate ganciclovir, it generates a metabolite that's toxic to dividing cells. And this was a way to inducibly eliminate glial cells in the gut. And what they found was that the phenotype was not subtle. These mice really developed fulminant hemorrhagic inflammation, particularly in the distal small intestine, that was associated with a very leaky, compromised epithelial barrier, altered crypt cell proliferation, as well as degeneration of enteric neurons. Subsequent studies by other groups using a variety of approaches to target glial cells in different ways similarly found this type of inflammation. And this really established a paradigm that the glial networks were essential for regulating the integrity of the intestinal epithelial barrier. And when these glia were disrupted, the barrier became leaky and caused inflammation to the host. One thing that was common to all of these studies was they all used the same glial promoter, the GFAP promoter, to drive glial ablation. And something that was recognized even from the first descriptions of GFAP expression in the gut by Jessen and Mirsky in the early 1980s was that not all enteric glia express GFAP. Wherein Bozeman's and colleagues in Vasilis Paknis's lab showed very nicely that even within my enteric ganglia, which are the most studied, that there were subsets of glia that were GFAP negative. We extended this work to really look at the broader population of glia outside the myenteric plexus and found that while almost all glia expressed S100 beta and PLP1, not all glia expressed GFAP, particularly in the mucosa as you go further in the distal intestine. So being particularly interested in the functions of what these mucosal glia were and how they might be interacting with the gut epithelium to regulate inflammation, we decided to target this wider S100 beta and PLP1 population for ablation and ask what happened to gut function. And to do this, we generated PLP1 pre-ER DTA mice in which we could inducibly express the cell autonomous toxin DTA or diphtheria toxin subunit A and ask what happened to gut function if we eliminated PLP1 expressing cells. And certainly in this model, in young adult mice, we were able to eliminate the large majority of glia throughout the small and large intestine. And importantly, we could see over 90% loss of the mucosal glial population that interacts most closely with the intestine. So of course, based on the previous work, we were really expecting to see fulminant inflammation. And in fact, much to our surprise, we saw no inflammation whatsoever. And not only did we not see inflammation, we saw no defects in the permeability of the intestinal epithelial barrier, nor in crypt cell proliferation. And even more remarkably, when we challenged these mice that were missing glia with a real inflammatory stimulus, DSS colitis, we saw that the mice fared no worse than their control litter mates that had glia. And this really suggested that in vivo, that enteric glia are not absolutely essential for maintaining the integrity of the intestinal epithelial barrier. So if they are not important for this, what are they important for? So we decided to look at gut motility, which is one of the most famous functions of the ENS. And again, we were in for a surprise. When we measured GI transit time in these mice, we found that in male mice lacking glia, there was no difference. And in female mice, remarkably, GI transit actually sped up. And this was associated with increased colonic contraction frequency, really suggesting that glia are essential, not for necessarily regulating the intestinal epithelial barrier, but they do seem to be important for regulating colonic motility, but in a sex-dependent manner. In support of this elegant work by Keith Sharkey's lab, as well as Brian Gulbranson's lab, in which they disrupted enteric glial function, but not necessarily depleted the enteric glia themselves, using a variety of approaches, they had similar findings that the barrier function remained intact. There was no increased evidence of inflammation, but there were defects in or changes in gut motility. And Brian's work in particular really highlighted the colon as a place where glia seemed to be particularly important for regulating motility. We were struck by this, as well as the remarkable sex dependence of this phenotype, which led us to wonder, well, there are so many GI disorders that have prominent sex differences. Is it possible that something as fundamental as the neuroglial circuits that regulate gut motility and peristalsis might be different between males and females? And in particular, what was surprising to us was that in the males, you could take away this huge population of cells and GI transit time and colonic motility remain remarkably intact, leading us to wonder whether male sex hormones or androgens could be playing a role. So the primary male sex hormones or androgens that circulate in male mice as well as humans are testosterone and a version of testosterone called dihydrotestosterone. And both testosterone and DHT signal through a single receptor, the androgen receptor, which is a nuclear hormone receptor. And studies in the 1980s by Winborn and colleagues, in which they radiolabeled dihydrotestosterone and injected it into animals, revealed that there are actually cells in the muscularis externa of the gut that could bind this ligand, suggesting that there are cells in the gut that were androgen responsive. Subsequent work by Mario Diaz's group in Spain showed very nicely that actually the visceral smooth muscle of the gut exhibits altered contractility in vitro in the presence of androgens. But it was unclear what effect or important functional significance this had in vivo. So to get at this question, we did a very simple experiment, which was simply to take wild-type postpubertal male mice and take away their gonadal androgens by performing bilateral orchiectomy or surgical castration or control operations, letting them fully recover, and then measuring their GI transit time 4 weeks later. And remarkably, we found that when you take away male gonadal function, GI transit time dramatically slows down. And this is really a fixed persistent deficit. So these mice don't get any worse over time when followed for several months, but they also don't get any better and they're unable to compensate. So to see if androgens were really the key signal from the testes that was important for this phenomenon, we performed same surgeries on a second cohort of mice and then actually replaced a single androgen, DHT, but in the form of a subcutaneous pellet. And what we found was that 1 week of DHT supplementation was not sufficient to rescue the phenotype, but by 3 weeks, the GI motility deficit had completely recovered. And this suggested that 1, androgens were sufficient to rescue the dysmotility, and 2, this was likely a genomic effect because it required such a long period of time. So then we'd asked what part of the gut might be particularly sensitive to the effects of androgens on gut motility. And we looked at this in mice, we looked at upper GI motility, gastric emptying, and small intestinal transit, and found that it stayed pretty intact, suggesting that the deficit was really in the colon. So to get at this, video is worth 1,000 words, and hopefully you can see this. This is a colon from a sham-operated mouse, where you'll see that a colonic contraction starts at the proximal end and progresses nicely along the gut. In contrast, you can see in an orchiectomized littermate that the contractions are highly disorganized and don't seem to be necessarily propulsive or productive. And this really suggested to us that androgens are particularly important for regulating motility in the distal intestine in the colon. So to understand which are the cells in the gut that might be responsive to androgens and responsible for this phenotype, we decided to look for androgen receptor expression in the postpubertal male colon. And what we found, as expected, was that there were lots of red androgen receptor immunoreactive cells throughout the smooth muscle. But what we didn't, what surprised us, that we also saw androgen receptor positive cells within the myoenteric plexus, where the signal co-localized with the pan-neuronal marker ANNA1. So, and remarkably, up to 20% of enteric neurons in the myoenteric plexus in the male colon are actually positive for androgen receptors, suggesting that they can respond to circulating androgens. And this is not true in the submucosal plexus. So the dysmotility that we observed upon depleting gonadal function in male mice could be because androgens are signaling to smooth muscle, they're signaling to enteric neurons, or potentially both, both, and these signals are important for motility. And in a lot of work that I don't have time to show today, but you can see in our bioRxiv paper, the muscle contractility actually remained remarkably intact, suggesting that the effect was actually due to these enteric neuronal androgen signaling. So to get at this with more specificity, we took a genetic approach and actually ablated androgen receptor expression in the peripheral nervous system using Wnt1-Cre2 mice, which is active in all neural crest derivatives. So in mice lacking the androgen receptor, specifically in their peripheral nervous system, what we saw was that before puberty in juvenile mice, that the knockout mice had the same exact GI transit time as their control litter mates. However, after puberty, these mice had markedly slow GI transit compared to their litter mates, very similar in magnitude to our surgical model. And so we think that circulating androgens produced by the gonads, such as testosterone and DHT, are signaling to at least two populations of cells in the colon. One's a smooth muscle, and one is a subset of enteric neurons. And this enteric neuronal androgen signaling actually seems to be critically important for the normal regulation of gut motility, suggesting that androgen signaling should really be considered as a potential cause of chronic dysmotility. And in further work also that you can find in this paper, we showed links to IBS pathophysiology. So hopefully over the arc of this talk, I've convinced you that we've learned a lot of interesting lessons by studying glia. One, that they're really a unique heterogeneous population of cells in the nervous system, and they have no direct analogy to other glial populations in the nervous system, suggesting that they really merit dedicated study. We started out thinking they were going to be critical for regulation of barrier function, but in fact, they seem to be dispensable for that, but critical for regulation of colonic motility. And I would argue that many of their homeostatic functions really remain to be fully defined, and that it's going to be essential to move beyond only studying myenteric glia to really also understand the functions of these diverse populations of glia throughout the gut. And hopefully the androgen story has given you some insight of what we're excited about, that really unbiased functional interrogation of these cell populations will really uncover new and unexpected insights on how the ENS plays a role in both digestive health as well as disease. And with that, I'll end and just thank the really fantastic members of my lab, and in particular want to highlight 4 people who are critical to the androgen work: Daniela Rosselli and Lindley Matthews, who really initiated and spearheaded that project, which is now being carried forward in a great way by Ari Robinson, Christina Perez, and more recently, Ballin. With that, I'll also acknowledge my collaborators, those who contributed reagents, as well as my funding sources. And I thank you for your attention.
Thank you, Meena. That was a superb talk, and really highlighted the role of these cells. Now, as I say to everyone in the audience, we were strongly encouraged to have the 3 talks back to back, and then question and answers at the end. So please put any questions you have for Dr. Rao or any of the other speakers in the question and answer. But without further ado, then we'll move on to our second speaker. And it's a pleasure to introduce Simon Gibbons. Simon's a research scientist and professor of medicine in the enteric neurosciences program at the Mayo Clinic. He studies cellular and molecular physiology of motility patterns in the GI tract, but he's got a specific interest in the role of interstitial cells of Cajal and innate immune cells in the establishment of motility in both health and disease. So, without further ado, we'll hand over to you, Simon.
Thank you very much, Keith. I appreciate the very nice introduction. The main points that I'm going to cover today relate to the distribution and significance of innate immune cells in the gastrointestinal tunica muscularis. Particularly, I'm going to focus on some of our work that's involved studying the role of gastric muscularis propria macrophages in an animal model of diabetic gastroparesis, and then briefly just discuss the potential clinical significance of what will primarily be our studies in mice. The presence of innate immune cells within the gastric tunica muscularis has been known for a number of years. These very early studies from Hannah Mickelson and Lars Spunerberg showed that many innate immune cells that were phagocytic cells were within the bioenteric region of the mouse small intestine. Here's another beautiful example from Bob Roberts and Terry Powley's lab showing the piling of the macrophages across the tissue against the background of the enteric neurons labeled with HUCD. In our own work, this is from a paper that we just published in Neurogastroenterology and Motility, we showed that the macrophages, as shown in green here, are located within both the myenteric region and the muscle layers in the small intestine. Then there's also macrophages on the serosal surface, and there are macrophages intercalated with the myenteric ganglia and adjacent to the deep muscular plexus, which I've defined here by using an antibody against the KIT receptor tyrosine kinase, which identifies interstitial cells of Cajal. In the stomach, it's a very similar story. There are macrophages within adjacent to the myenteric region, and there are macrophages that are adjacent to ICC throughout the muscle layer. And in the stomach, they really are throughout the muscle layer. If one looks at their relationship to other cells, zooming in a little bit on their relationship to nerves and nerve fibers here, in this beautiful work from Gianluca Cipriani, a postdoctoral fellow in our lab, currently developing his own programs. Again, you can see here how closely associated the macrophages are in the smooth muscle layer and the myenteric region. And they also appear to be reasonably close to the PDGFR alpha positive cells, also known as fibroblast-like cells, and or recently nomenclature of telocytes has been coined by some investigators. So how does this look if one looks at it in 3 dimensions? I'll show you a video here of MHC2 positive macrophages in the small intestine in the myenteric region. What I think is particularly interesting is how fine the processes are of these macrophages. They are, in some ways, almost appear to be penetrating the tissue, and they're running very closely adjacent to the nerve fibers, maybe only a few tens of nanometers thick. Even in the muscle region, then what one can see is that, again, the nerve fibers here are labeled with PGP 9.5, and the macrophage processes are located quite exquisitely adjacent to the nerve fibers. But the real test for whether something is adjacent or actually touching, given the limitations of light microscopy, is to zoom in on electron microscopy. And to that, we've gone to the most skilled people in this field, Simonetta Fassone Pellegrini and Maria Giuliana Vanucchi. And what one can see, what we found when we looked in both mouse and human, and there are examples here from this recent paper that Gianluca Cipriani led, that the macrophage is very close to the myenteric plexus. I mean, I've already said that, and there's no reason. But what's interesting is that there's almost invariably a gap of a few hundred nanometers, whereas when the macrophage is adjacent to a smooth muscle cell, one sees seaming membrane specializations in areas of contact. In this case, what looks very much like a peg and socket type relationship. And again, here there's these membranes are essentially inseparable. They're forming contacts with smooth muscle cells. They're also forming contacts in this image here from the muscle layer and the colon. Here we're in the small muscle. Here we're in the small intestine. There's the myenteric plexus region, but all along this line of small red asterisks, there's a gap there. The macrophages do appear to form close contacts with the PDGFRα or telocyte fibroblast-like cells. They do appear to form specialized contacts with the interstitial cells of Cajal. But here in the myenteric region of the colon, there doesn't appear to be any specialized contacts with nerves. Here we have the very fine process, which is characteristic of the telocyte in a human stomach, right next to an identified muscle macrophage. So what we've got here is we've got cells that are forming a variety of different relationships with other cells in the muscle layers of the tissue. And what this really illustrates is that when it comes to innate immune cells, we're way beyond the point where the old model had classically activated M1 macrophages that mediate immunity and alternatively activated macrophages that mediate cytoprotection. There's a spectrum going on. And many of the people, there have been many investigations in the field that demonstrate that they have diverse phenotypes and functions. Some exquisite work from Dan Maceda's lab in Rockefeller showed the relationship in terms of using BMP2 to support survival of neurons. We did quite extensive studies showing that some macrophages appear to generate IL-10 and carbon monoxide that appear to protect interstitial cells of Cajal from injury. We also demonstrated that separately, some macrophages are producing factors such as TNF-alpha and IL-6, which are leading to dedifferentiation and injury of ICC. We know that macrophages present antigens and are professional antigen presenting cells with MHC2. We also know from work from Subhash Kulkarni that they are clearing debris and phagocytosing neurons. One area that hasn't been investigated in our field is whether they may also play the same role as myeloid cells in the brain as far as modulating synaptic plasticity. Indeed, the actual relationship between macrophages and motility in different conditions has been investigated by a large number of groups. Lauren Becker and Ida Habtazian's work showing how age was shifting the polarization of macrophages to more pro-inflammatory phenotypes. Hugh Boxteen, Gianluca Mattioli, and the people at Leuven have been looking at postoperative ileas for many years and looking at how CCR2-dependent monocytes might resolve inflammation. Here's some work from Dan Maceda looking at the relationship between infection-induced neuronal loss and the modulation of macrophages by the extrinsic nervous system through adrenergic signaling, and Hong-Jane Hu's work on the TRPV4 signaling on through muscle cells. What we've been looking at is diabetic mice. And we've used two types of type 1 diabetes models, the NOD mouse that reliably develops diabetes and the Black 6 and genetically modified strains, which we induce using streptozotocin. The fact the fundamental way that principle for these studies has been our ability to measure gastric emptying of solids in a reliable way in non-restrained animals. And a subset of the mice do develop delayed gastric emptying. The critical factor appeared to be the loss of the KIT ICC marker and damage to ICC networks in the diabetic mice that had delayed gastric emptying. And the other factor that we found in these studies now some 10 years ago was that whereas there was a modest increase in the total number of macrophages identified here with F480, what was striking was that in mice with normal gastric emptying that had been diabetic for 10 weeks, there was a big increase in the proportion of these cells that was positive for CD206. These cells were also positive for hemoxygenase 1, which generates carbon monoxide, a powerful cytoprotective factor in tissues. Despite its broad systemic toxicity, it does have some positives. And what is clear here, this is an example here of a diabetic mouse with normal gastric emptying. And this is a mouse that had developed delayed gastric emptying, usually between 4 to 6 weeks after the onset of diabetes. So our hypothesis at this point was that the failure to sustain this expression of anti-inflammatory macrophages leads to diabetic injury. But what surprised us was when one looked at mice that didn't have macrophages at all, that we didn't observe delayed gastric emptying. And to do that, we used a mouse model, the CSF1 OPOP mouse, which doesn't generate functional CSF1 and therefore does not appear to have more than one or two, this occasional macrophage in the tunica muscularis of the stomach. The wild type mice of this strain develop delayed gastric emptying, or about 40% of them do. But the mice that are made diabetic that do not have macrophages do not appear to develop delayed gastric emptying, even in the presence of quite significant diabetes. So what happens when we put macrophages back into these mice? And by administering CSF1, we were able to observe that there was population of the tunica muscularis with F480 positive macrophages. And in those mice that were made diabetic, then given CSF1, then the vast majority of those mice developed delayed gastric emptying. They also developed the characteristic cytopathological signs of damage to the ICC networks in the mice with delayed gastric emptying. So what we appear to have is that we've got gastric emptying in diabetic mice being modified by the generation of protective and injurious factors in diverse myeloid cells. We've got hemoxygenase 1 producing antioxidants. We've got antigenase 1 breaking down the source of nitric oxide. We've got we've got the production of interleukin 10. On the other hand, in mice with delayed gastric emptying, then the most abundant factors appear to be production of TNF and IL-1 beta, upregulation of INOS or NOS3 in mice, but not humans, and IL-6. So somewhere in this spectrum of macrophages, there are cells that are producing some or all of these factors. And where are these macrophages coming from? Well, really, it comes back to some elegant work from Leuven again, where they showed that, like many other tissues, the macrophages within the muscularis of the small intestine, at least, are being populated during the embryonic period by yolk sac-derived cells, but just like microglia establish themselves about halfway through gestation. At the same time, there are cells that are populating the tissue that are being derived from monocytes, in the embryo from the fetal liver, and then in the adult from monocytes from bone marrow-derived macrophage and dendritic cell precursors. And these cells, these monocyte cells, appear to be able to turn over, and the yolk sac cells become long-lasting, self-sustaining cells. So how are we going to characterize this? Well, one of the problems here is that our traditional tools of immunohistochemistry and flow cytometry have some limited power to discriminate subtypes of muscularis propria macrophages. For example, here we've got a couple of classical markers of macrophages, MHC2 and CX3CR1. And the CX3CR1 should be expected to be a subset of the MHC2 positive cells. But there are evidently CX3CR1 cells that are negative for MHC2. They're not antigen presenting, but they have this macrophage-like phenotype. Similarly, not all of the F480 positive cells are also not all of the CX3CR1 cells are also F480 positive. And indeed, using these two widely used markers for macrophages, when you do a flow cytometry on associated small intestine, for example, in this example, what one finds is that about 20% of the antigen-presenting cells are negative for F480. About 20% of the F480 positive cells are negative for the antigen-presenting cells, and about 20% of the antigen of the F480 positive cells are also negative. So the way that we're approaching this is we're doing what many people are doing, which is to use the tool of single-cell RNA sequencing. When we dissociate our tissues, we find that the cells, based on their transcriptome, can be classified into multiple clusters. Of these clusters, the MHC gene, 2 genes fall into this cluster here. A subset of these are CX3CR1 positive. And of the CX3CR1 positive, we find that this cluster here, shown in the blue circle, is significantly enriched for 8 of the 9 genes, which have been characterized by Schipper and colleagues as representing the long-lasting self-sustaining macrophages, or the microglial genes. So obviously, the ongoing studies are going to take this much further, and time prevents me from really going more into the details of this. But I do want to point out that we feel that these mouse studies, which have informed a lot of the work of the GPCRC, are very significant. My colleagues Madhu Grover and Gianrico Ferrugia, working with the GPRC, have published a series of papers of which I've been a part, but initially, we demonstrated that cellular changes in diabetic and idiopathic gastroparesis matched what we were seeing in mice in terms of loss of interstitial cells of Cajal and increase in immune cells, that the loss of ICC correlated with the loss of CD206 positive macrophages. And when looking at the transcriptome of the tunica muscularis from biopsies, that many of the macrophage-associated immune pathways, such as atherosclerosis signaling, show up as being significantly upregulated. And it's always the proteins that do the work. So a proteomic profiling found 40 common macrophage-associated proteins in patients who had gastroparesis. So to conclude, development of delayed gastric emptying in diabetic mice, an animal model of diabetic gastroparesis, depends on a balance of cytoprotective and injurious factors in macrophages in the gastric muscularis propria. It's likely that the origin and environmental exposures of subtypes of muscularis propria macrophages contributes to phenotypic changes in response to diabetes and immune cell activation patterns associated with gastroparesis in people. I have a huge list of acknowledgements, and I'm not going to go through this in detail. I do want to thank the NIH for support of the GPCRC in particular, and the NMS for the support of a Young Investigator Award to Dr. Cipriani. And with that, I'll pass back to Dr. Sharkey. Thank you.
Thanks very much, Simon. It's a great talk. So we're pretty much on time, but we'll move along here with our last speaker, Dr. Kenton Sanders, Professor and Chair of Physiology and Cell Biology at the University of Nevada in Reno. Ken Sanders has studied visceral smooth muscles and interstitial cells and their regulation of smooth muscle cell function for quite a few years now. His lab and his collaborators use a variety of techniques to really understand the specific role of each cell type in the gut and the generation of intrinsic electrical activity and indeed movement in the gut. And today he's going to highlight the functional connectivity between smooth muscle cells, interstitial cells of Cajal, PDGFRα cells in a syncytium that drives normal GI motility. So over to you, Ken.
Okay, I hope thank you very much, Keith and Bev, for inviting me to give this talk. I'm going to get right on to it because it's the time is short. The first thing that I want to have people consider is the importance of the morphology of interstitial cells, not only in relationship to the smooth muscle cells, but also neurons. In the gut, typically these bundles of neurons are found, and there are close associations between both the PDGFRα positive cells and the ICCs. In the case of ICCs, you can find very close apposition between the varicosities of enteric neurons and the ICCs, in this case, only about 20 nanometers. And these can be found fairly commonly in the tissue. There are occasional close junctions with smooth muscle cells, but when this was done with morphometrics years ago by Ed Daniel, the closeness of varicosities to ICCs was remarkable. Now, it's true that when neurotransmitters released into a narrow gap like that, high concentrations are reached, and therefore it may be a site of neurotransmission as was proposed by morphologists. Another really important feature is that ICCs and PDGFRα cells are connected to smooth muscle cells by gap junctions. Here's an interstitial cell that was done by Professor Komarov's lab, and a PDGFRα cell the same way, a neural bundle close, and then PDGFRα cell with a gap junction to the smooth muscle cells. So the point of this is that when ion channels are activated in PDGFRα cells or KIT positive ICCs, it's possible for currents to be conducted from one cell to the other so that these cells can influence the behavior of the smooth muscle cells. A little map that I've drawn up just so that you can put it in your mind is that there are cells within the ganglia regions of the gut that contain multipolar PDGFRα cells and ICCs. There are also cells in close contact with the intramuscular projections of the enteric nervous system. We call these intramuscular ICCs and PDGFRα positive cells. Now, we've spent quite a bit of time dissecting the function of isolated cells from tissues. We've learned to disperse the cells, and then with reporters to find the cells in the dispersions. And basically, this summarizes the functions of the cells and the important receptors and effectors that are present. Smooth muscle cells, of course, develop force. They're responsible for the forces of motility. They also respond to a whole variety of bioagonists and have receptors for a number of things. Pacemaker activity and responses to bioagonists are present in these multipolar type of cells in pacemaker regions. These cells express receptors to many bioactive agonists. In fact, we're still finding out about new agonists that affect ICCs. They have prominence of ryanodine and IP3 receptors, so a role in calcium signaling. They also express T-type calcium channels, which is very important for propagation of activity, as I'll show you. And then finally, ANO1 channels, which are that respond to calcium signaling. They're a calcium activated chloride channel. The intramuscular type of ICC, which are typically called ICC-IM, in the small bowel, they reside in the deep muscular plexus, so they're often called ICC-DMP. And there's also a group of cells along in the subserosal region of the colon. These cells exhibit tonic, excitatory, or inhibitory tuning of the syncytium of cells. They have responses to excitatory neurotransmitters such as acetylcholine and neurokinins, and inhibitory responses to nitric oxide. They have expression of neurotransmitter receptors such as M3 and soluble guanylate cyclase. That's the main cholinergic receptor and the receptor for nitric oxide. And in the small intestine, they have receptors NK1 for tachykinins. Again, they also have ryanodine IP3 receptors and ANO1 channels. And then finally, the PDGFRα cells exhibit tonic inhibitory tuning in the colon particularly. They show responses to purines, and recently we've shown very important responses to catecholamines. These cells have neurotransmitter receptors P2Y1 predominantly, and the alpha-1 adrenergic receptor that gives them responsiveness to catecholamines. They also express SK3 channels. Now, a fundamental behavior of all ICCs throughout the gastrointestinal tract is calcium signaling. In this little movie I'm going to show you, we've looked at spontaneous activity in ICC-DMP, and these events go on continuously in cells. In the second panel, one cell has been made blue, which is used, we've made this in the vertical axis, and then created spatiotemporal maps, which show all the different sites of calcium release. And in this movie, you can see spontaneous calcium transients going off in these cells. And in the spatiotemporal map, you can see that there are many sites. This is stochastic activity. It goes on continuously, but there's no relationship between the activity site to site in a given ICC or in adjacent cells. We've seen no evidence for what has been called, coupled oscillators or calcium phase waves and that sort of thing in these cells. The calcium transients are quite discrete and independent. The reason that calcium transients are important in ICCs are because of the expression of these ANO1 channels, which are calcium activated chloride channels. This is a patch clamp experiment showing the spontaneous transient inward currents that occur at physiological potentials. They reverse at 0 when ECL is 0 millivolts, and they reverse at minus 40 millivolts when ECL was set to minus 40 millivolts. These spontaneous transient chloride currents going on in many, many cells throughout the tissue provide a net excitatory input. There are also spontaneous transient calcium transients in PDGFRα cells. However, the ion channels in those cells are SK3 channels. And so calcium couples to the activation of a potassium conductance and produces outward currents that are blocked by apamine, therefore SK type currents. If you look at the contributions of all of these channels and effectors in the SIP syncytium, this little dissection was done very nicely by Sean Ward in this experiment, where he took a small bundle of smooth muscle cells that of course would contain ICCs and PDGFRα cells. These bundles of tissue produce spontaneous action potentials. Those events occur in smooth muscle cells, and they're due to L-type calcium channels. They're blocked by nifedipine. What's left in the presence of nifedipine is a noisy baseline where there's sort of random fluctuations up and down. And when a chloride channel blocker is used that blocks ANO1, then we're left with just downward deflections. The downward deflections are inhibited by apamine. So you've got spontaneous contributions from all of these different cells in different ion channels and different roles in the tissue, but the overall effect is a tuning of the excitability of the tissue. And a good example of that is we ask the question of why the membrane potential in the colon is so depolarized relative to many other areas in the gut. And sure enough, benzbromarone, another calcium activated chloride channel blocker, caused a significant hyperpolarization. So in other words, the spontaneous activity of ICCs going off causes a net sort of depolarization or excitability in the smooth muscle cells, upon which all the other regulatory mechanisms are superimposed. So just to summarize this basal activity, stochastic release of calcium in ICC and PDGFR alpha cells is ongoing. Calcium transient causes activation of ANO1 channels in ICC and SK3 channels in PDGFR alpha cells. Currents from ANO1 channels elicit spontaneous transient depolarizations in ICCs. Currents in SK3 channels elicit spontaneous transient hyperpolarization in PDGFR alpha positive cells. The STDs and STHs together conduct to electrically coupled smooth muscle cells and contribute to setting the basal excitability of the subsyncytium. Okay, so now I mentioned the proximity of ICCs to enteric neurons, and here's an example of stimulation of neurons with transmural nerve stimulation in favoring nitrogic input to the tissue. And again, in this movie, we have a differential image really shows up the calcium transients nicely that are going on in this stochastic manner, as I was mentioning. In this panel, we have just the total number of pixels, the active area in the tissue, and then an analysis of particles. When the stimulator's turned on, you can see that the calcium transients just die out briefly during nerve stimulation. And then after nerve stimulation, they sort of go crazy a little bit, this sort of an after excitatory effect that has been known in gut muscles for a long time. So in inhibition of calcium transients by nitrogic stimulation, I don't have time to show all of this, but this is completely blocked when nitric oxide nerve stimulation is blocked. And excitatory input produces exactly the opposite. So there's some important basic principles in motor innervation that I'm not going to cover in great detail because of time. Motor neurons regulate the calcium release events in ICC and PDGFR alpha cells. Excitatory neural inputs, which are cholinergic and peptidergic, increase calcium release, and inhibitory inputs, which are nitrogic, decrease calcium release in ICCs. The inhibitory neural inputs, which are purinergic and sympathetic, increase calcium release in PDGFR alpha cells, which of course I didn't show. Calcium release is linked to activation of ANO1 channels in ICC and SK3 channels in PDGFR alpha cells. So enhanced calcium release increases ANO1 openings in ICCs and produces a depolarizing or excitatory trend. Enhanced calcium release increases SK3 openings in PDGFR alpha cells, causing a hyperpolarizing or inhibitory trend. Inhibiting calcium release in ICC reduces basal excitability and causes a hyperpolarizing or inhibitory trend. And the ongoing activity of nitrogic neurons in the proximal colon inhibits basal calcium release events. And this is the basis of what's known as tonic inhibition in the proximal colon. Now I just want to move on to the cells that activate slow wave activity, rhythmicity in the gut. It's a beautiful picture provided by Teramasu Komuro of the ICCs in green and the enteric nervous system in red in the myenteric region of the small intestine. And what we found several years ago was that calcium events traverse this network of ICCs and produce, and there are in association with slow waves. When recordings are made simultaneously with slow wave activity and calcium transients or global calcium activity in these networks, you can see a 1-to-1 correlation between the two. And careful observation at high power shows that these global calcium transients are not whole cell calcium events, but in fact, they're quite specialized and quite regionalized into specific points of release. This particular slow wave is blown up on the next panel. And you can see that the event that's propagating through the tissue, this is the upstroke of the slow wave that occurs. And then shortly after that upstroke event occurs, you start to see transient calcium release events going on at all these different sites within cells, within the network of ICCs. So the calcium events that are occurring normally in ICCs are clustered by the calcium action potential that essentially occurs within the ICC. And this organizes the calcium release events into clusters. A very dramatic example of this is in gastric ICCs. We wondered whether or not the duration of calcium release events would be increased in these preparations because they have long slow waves. And sure enough, when this was looked at, we saw hundreds of calcium release events within a network of ICC, and then a quiescent period. And then you begin to see calcium events pick up right before the explosive event that occurs as the slow wave. And so you have calcium transients contributing to the pacemaker activity, the onset of the slow wave, and you also have calcium transients contributing to the duration of the slow wave, the plateau event, which of course is due to activation of ANO1 channels. In the absence of ANO1 channels, there are no slow waves that develop in either the small intestine or gastric antrum. So to summarize these principles, stochastic calcium release events in ICC are organized into clusters by voltage-dependent calcium entry. Expression of voltage-dependent calcium channels defines the difference between ICC and the muscle layers that generate, just calcium transients and spontaneous transient inward currents, and cells that generate slow waves, ICCMY, ICCSM. Calcium activity, calcium entry through voltage-dependent calcium channels initiates the depolarization and causes calcium-induced calcium release. This initial depolarization is the upstroke of the slow wave, and the period of calcium-induced calcium release is the plateau phase. Calcium-induced calcium release persists for a second or more, and the duration and magnitude of the clustered calcium release events defines the amplitude and duration of the slow wave plateau. That's important because that's where major motility or major contractile responses occurs. Calcium release and activation of ANO1 channels contribute to the two phases of slow wave activity in this way. Pacemaker events that initiate slow waves, those are the events that occur just before the upstroke of the slow wave, and generation of the long duration plateau phase that predicts that predicated actually the term slow wave. So this is my final slide, and it seems really complicated, but I'm going to go through it really briefly. The basic events of ICCs and PDGFR alpha cells are calcium release. Calcium release is modified by neural inputs. I've shown here the excitatory input and the inhibitory input. Inhibitory inputs through PKG-driven mechanism inhibit calcium release. The excitatory inputs increase calcium release. In PDGFR alpha cells, which I didn't have time to really go into too much, P2Y1 receptors cause an increase in SK3 channels activation, and so does cholinergic input from norepinephrine release from sympathetic nerves acting through adrenergic alpha 1 receptors. ICCs also produce slow wave activity. They have spontaneous calcium transients, but these events are organized into these clustered events that I showed you to produce the slow wave activity. both the events from ICC-IM and PDGFR alpha positive cells and ICC-MY conduct to smooth muscle cells, producing the behaviors of motility through activation of calcium channels and activation of the contractile element. There are also direct neural activation of effectors in smooth muscle cells that can regulate the calcium sensitivity of the contractile element. Together, these cells we call the CYP syncytium for smooth muscle interstitial cells and PDGFR alpha cells. After what Simon told us tonight, we might need to add macrophages, and I guess this would become the CYPum syncytium. That's all I have, Keith.
Thanks very much. That was great. So I'll ask the speakers to put their video on now and their microphone so that they can answer some questions. We're a little bit behind time as I see it here, and I hope the organizers will let us go on for a few minutes after the hour. I've got about a dozen or so questions that have come up in the question and answer. If any of these questions are not answered, the speakers will be asked to write an answer, and there'll be a slide at the end of the presentation here where you can note down the URL and you can go on and get answers to those questions. So I'm going to start with a couple of questions here related to Meena's talk. And the first one from Brian Galbranson, and a very similar question also from Jack Grider, is asking you the 20% of neurons in the myenteric that express androgen receptors, are they any specific subtype, or is it mixed amongst the different subtypes of neurons?
So far, from what we can see, it looks somewhat mixed. So definitely a substantial proportion of them seem to be NOS and VIP positive, suggesting they're probably inhibitory motor neurons, but definitely not all. And that goes both ways. So not all the NOS neurons are androgen receptor positive, and not all the androgen receptor positive neurons are NOS positive. But that seems to be one major population.
Fantastic. Dr. Shashevsky's asking you, Meena, have you done ovariectomy and looked at colonic motility?
Great question. I feel like a traitor to my sex and gender, I guess, for not having done this. But actually, part of why we started with androgens was also because there's one receptor, and it's definitely a less complex system to study first. We haven't yet done ovarectomies, but that's definitely something we're very interested in and about to embark upon.
Fantastic. Thank you. A question now also related to androgens from Bard Ermentrup. Is it known why the lack of androgens in the castrated mice caused the disorganized motility patterns?
That's definitely $1 million question for us. We're very interested to understand it, and I don't have sort of an aha answer to give right now, but definitely an area of active interest. We're trying to understand whether it's affecting, AR is a nuclear hormone receptor that affects gene transcription. So we're trying to understand if there's certain programs that are being altered that might be playing a role.
Great. Well, I'm going to move on to a couple of questions for Simon now. Brian Galbranson asks, based on the single cell sequencing profile that you showed, it seems like there are some macrophages that are linked along a spectrum, while others may be quite different and not clearly linked to the others, so some separate groups. Do you have any additional insight into transitions between the macrophage subtypes or developmentally different subtypes there, Simon?
We really started to look into that, and I think that's the reason for that's really the reason for why we started taking this approach of looking at single cell transcriptomics. The CX3CL1 cells, we've looked at those by tagging them with green fluorescent protein, and they are diminished in diabetes. But whether it's one of those two subsets or the other that's being affected by the onset of diabetes and changes in gastric emptying, we're not sure yet. For that, they're doing some lineage tracing experiments.
Right. Great, thanks. And then Dr. Kulkarni is asking you, how does one reconcile traditional ICC markers, ANO1 and KIT expression by their seeming expression by myenteric neurons, according to the recently published single-cell RNA-seq data of the enteric nervous system?
So I'm going to profess that I haven't been doing my homework, and I haven't really looked at those data really closely. Obviously, I'm aware of both those two excellent papers that have come out from Harvard and from the Karolinska. I can tell you that we've looked very closely at whether or not ANO1 is expressed in other cells. The functional protein is certainly not expressed in any meaningful way in cells other than ICC in the muscle layer. And when it comes to KIT, the same is true. There's plenty of evidence from using drivers from the Kent generated the KIT cop GFP mouse, which gives us a very clear idea that any signal of transcriptional signal that's showing up in single cell, single cell RNA-seq is not the deepest sequencing technique. It's a really powerful technique. We're really working to learn it more. Combined with things like a single cell attack-seq and other stuff, then we can learn a lot. But I'd just say, let's look at the preponderance of more than one gene mark. And the beauty of single cell is you can do that. You can look at 3 or 4 things and say, this cell type is clearly one or another, or this cell is clearly not different. That's where we rely on biostatisticians.
Yeah, thanks for that. Then we have a question from Bianca Zassino. Would stem cell therapy be beneficial in correcting gastroparesis by creating more ICCs and boosting the immune system?
I think there are a lot of people that love to think that's the case. It's not an area that I've looked at. So I would, yeah, it's feasible. A few people have looked into it, but it would be speculative at this point to think that it might make a difference.
Right. Well, thanks, Simon. Over to you now, Ken, and your former mentor, Dr. Krzyzewski, wonders, is activity in the PDGFR alpha cells and ICCs synchronized? And if so, what is the basis of this synchronization?
As far as we can tell, there's no synchronization between the activity in ICCs and PDGFR alpha cells. They, in fact, even between 2 ICCs and even between 2 calcium release sites, a few microns apart, there doesn't appear to be synchronization of the basic calcium transient events without a slow wave-like event. Now, that being said, Joe, I should also point out some work by Yoshi Kito, where this is from the rabbit small intestine, where he recorded from both ICCs and then in separate experiments recorded from PDGFR alpha cells. And because this is a syncytium, he could record large amplitude slow waves in ICCs, but he also recorded attenuated small amplitude slow waves in PDGFR alpha positive cells because they're conducted there. So there's a possibility that current entering PDGFR alpha cells could have some effect, but at this point in time, that simultaneous measurement of calcium transients in both cell types has not been made.
That's great. Thanks, Ken. Last question then, in the interest of time, we have to end in a couple of minutes here, but last question to you, Ken, relates to the role of nerves, really. Just exactly, so the question really is, I'm trying to paraphrase it quickly, is just how important is the role of nerves in muscle activity relative to these other cell types in initiating propulsive motility?
Yeah, well, so we believe that most of the response in the intact muscles is mediated through ICCs or PDGFR alpha positive cells. we've shown that the responses of PDGFR alpha cells precede the response in smooth muscle cells. And in fact, smooth muscle cells do not even respond to purines in a manner that would be consistent with post-junctional responses. Similar experiments have been done multiple times on ICCs. And then with imaging now, we can actually see the response right in front of us when turn the stimulator on, that the response in ICCs is immediate along the lines of the delay between nerve stimulation and post-junctional membrane potential changes. So we know that responses are set up in interstitial cells, and when interstitial cells are missing, the responses are different. They're not completely gone, but they're different.
That's great. Well, I'd like to end there. I want to thank the three speakers for doing a marvelous job. There's some chat comments that say the same. Want to thank the ANMS for giving us the opportunity to share this really exciting area of research, the role of non-neuronal cells in the regulation of motility, which is what we focused on, but I think it's going to be clear there'll be other activities. I think the last slide that's going to come up, thank you, Stephanie, relates to any unanswered questions. My apologies to those who asked questions that were not answered today, but in the interest of time, we need to wrap up now. I would like to thank the ANMS for sponsoring this whole symposium series. Thank you as an audience. I noticed that a vast majority of those who started are still with us, and it's a great pleasure to have so many of our colleagues participate in these events. So on that note, I'll wrap up and say thank you, everyone, and have a very safe and happy holiday season.